Welcome! Thank you for watching the thawing and establishing of neural stem cells tutorial and also for using our products. Thawing and establishing of mouse neural stem cells. Materials required: a centrifuge tube rack, tissue culture flasks, a water bath, semi-dry alcohol swabs, a pipette, a nitrogen container, a centrifuge, 15 ml conical tubes, cryovials, a tweezer, a CO2 humidified incubator, neural stem cell growth medium. Thawing and establishing procedures. Pre-warm the NSC growth medium to 37 degrees Celsius. Add 9 ml of NAC growth medium into a 15 ml conical tube. Remove the cryovial of NACs from the liquid nitrogen container and quickly place it in the -80 degrees Celsius freezer. After 2 to 3 minutes, take out the cryovial of NACs from the freezer and quickly thaw cells in a 37 degrees Celsius water bath until the last bit of ice has melted. Note: be careful not to submerge the entire vial. Finish the thawing procedure as soon as possible. Maximum cell viability is dependent on the rapid and complete thawing of frozen cells. As soon as the cells are completely thawed, disinfect the outside of the cryovial with 70 to 75% ethanol. Use a pipette to transfer the cells to a 15 ml conical tube containing 9 ml of NAC growth medium inside a biosafety cabinet. Be careful not to introduce any bubbles during the transfer process. Rinse the vial with 1 ml of medium to reduce cell loss. Then transfer the cell suspension into the conical tube. Centrifuge at 140 times G for 4 minutes. Carefully aspirate as much of the supernatant as possible. Resuspend the cells in 1 ml of NAC growth medium pre-warmed to 37 degrees Celsius. Gently pipette up and down. Seed cells in a T25 flask at the density of 1 to 2 times 10 to the 5 cells per ml. Add a sufficient amount of NAC growth medium and gently rock the culture flask to evenly distribute the cells. Incubate cells at 37 degrees in a 5% CO2 humidified incubator. The next day, change spent medium with fresh growth medium. Changing the growth medium. Note: NACs tend to grow slowly after thawing. The growth medium should be changed when the neurospheres have formed but are not quite ready for passaging or when the medium turns yellow. Observe cells under the microscope. Be careful not to flap the bottom of the culture flask to avoid cell detachment. Note: avoid repeated warming and cooling of the medium. If the entire content is not needed for a single procedure, transfer only the required volume to a sterile secondary container. Transfer the cell suspension containing neurospheres to a 15 ml conical tube. Centrifuge at 140 times G for 4 minutes. Aspirate and discard the supernatant. Resuspend cells in 1 ml of NSC growth medium. Be careful not to disperse the small neurospheres. Transfer the cell suspension to a new T25 flask. Add a sufficient amount of NSC growth medium. Incubate the cells at 37 degrees Celsius in a 5% CO2 humidified incubator. The next day, change spent medium with fresh growth medium. Thank you for watching this tutorial.